Welcome to the GEOTOP-A lecture series. Our speaker this morning is Chad Giusti from the University of Delaware. He's going to tell us about tracking topological features across neural stimulus spaces. Chad, take it away.
Okay, thank you very much. And thank you to the organizers for the invitation and everyone else for uh, taking the time to join us here today. So I'm going to talk a little bit about how a method for using tools from applied topology in the context of studying how the brain encodes information. So I know that the intersection of people who study neural coding and algebraic topology is quite small. So I'm going to try to make the neuroscience as transparent as possible. A lot of these ideas are very recent, so they're also sort of new in the neuroscience world. So I want to start by just giving you some context in terms of the neuroscience. And I've got to make sure that I can click on a slide here. There we go. So I'm going to study these things called stimulus spaces, and I want to give you an idea what a stimulus space is. So this is a very classic video in neuroscience. Hopefully you can all hear the audio. There's some clicking going on. What's happening is that the people who are running this experiment in the 1950s have inserted an electrical probe into the brain of a cat in the primary visual cortex. And the clicking noise is a single neuron spiking. So it's activity on one neuron in the cat's brain. And what you can see, they're marking this location where if they shine the light, the neuron is active. And if you miss that spot, then the neuron is not active. Similarly, they're going to show you that there's a specificity to it, that if you have a very wide bar, you get some activity away from the main spot. And what they're discovering here is that there's sort of a Gabor filter at work. There's orientation sensitivity in addition to location sensitivity. So I'm not going to play you the whole 3-minute video, obviously, but there's something happening here. This neuron is active based on a stimulus out in the real world, and it's localized. So, right, there's now demonstrating orientation sensitivity. So the neuron doesn't fire if you don't have the right orientation of the edge in addition to location. So this is a fundamental result in the study of neuroscience, of cellular neuroscience. The people who did this experiment, Hubel and Wiesel, called these things simple cells. So there's a class of neurons in your primary visual cortex, which are what we call tuned to respond preferentially to both location and orientation of a stimulus on the retina. Okay, so this is a class of cell that lives in your brain that does some sort of coding for a stimulus. Much more recently, well, not much more recently, this is now in the 1970s, although this experiment won't be, this experiment's much more recent. A neuroscientist named John O'Keefe did some experiments. So now we're not going to hear the neurons spiking because there are 10 neurons, but they're each a different color. And you'll see the pattern emerge pretty much immediately. So this is an animal moving in its environment. We're recording from 10 distinct cells in its hippocampus, which is an area of the brain known because of these experiments to be involved in spatial location. And what you see is that these 10 neurons are each preferentially tuned to a location in the animal's environment. So again, we have some sort of localization of activity in terms of some behavioral or stimulus correlate. So this is John O'Keefe's work in the 1970s. He discovered this class of neurons now in the hippocampus, not the primary visual cortex, which we call place cells, and they're tuned to an animal's location during movement. Okay, and this is a very simplified picture. If you ask neuroscientists, they'll tell you there's a lot more here, but the idea is that there's this localization. One more, and this one's pretty cool. So this is much more recent work. This is another animal in its environment, and now we're recording from a place called the entorhinal cortex, which sits essentially directly above the hippocampus. There's a lot of interaction between the two. So place cells and the cells that we're viewing now are their activity is related in some way. And now we're just recording a single cell. So this is not obviously spatially localized, right? There's a lot of activity around the box, but we're going to speed it up here a little bit, and we're going to see a pattern emerge nonetheless. I'll just give it a second to play out. But again, this is activity from a single neuron in the brain, and hopefully that that pattern is a little more obvious now. So what they've discovered is that these so-called grid cells tune to positions in a hexagonal lattice. So if you were to take a hexagonal fundamental domain and overlay it on the environment, you would get periodic coding in that lattice during the animal's motion in the environment. Okay, so stimulus spaces, I claim these are all examples of this thing I want to call a stimulus space. So in the first case, we have response of the neurons localized to position and angle, and if I were a topologist, I'd say something like, a plane cross RP1, right? That would be my notion of what that stimulus space is, and then activity is localized in that space. Place cells or positions in the environment, whatever the environment happens to be. If I put you in an environment with a lot of different obstacles in it, you'll get, some sort of punctured plane. And the grid cell looks like a whole bunch of points, but of course, one of the first things that you do to your topology students, if you're a topologist, is you say, well, take a, let's build spaces by folding them up. So here's my, cartoon version of that that stimulus, or the receptive field for that neuron, the place where the neuron is active, along with an overlay of the hexagonal lattice, and I can, of course, pull out the fundamental domain and the gluing rules, and then I can fold and glue, and I can fold, twist, and glue again, and I get this nice torus with a twist in it. So there is a localization going on here. It's just not something flat. It's sitting in some kind of manifold. And the Moser Lab, who are the people who originally discovered grid cells, along with a few topologists whose names you might see sprinkled in there, published this very nice paper in Nature earlier this year, which shows that you can in fact find the torus sitting in the activity of these grid cells using tools from applied topology. They use a version of cohomology to do this. So it's in there, and we can find it, and we can characterize it. So this is position on a torus. So okay, so what is the unifying principle here? So this is the basic definition that we want to work with. A population of neurons has a stimulus space coding scheme if I have some metric stimulus space. So the neurons are going to be points in that space. So you can put you embed the points as neurons as fixed points in the space, and then if I stimulate if I sort of give a stimulus or an input to the system, that's going to correspond to selecting a point in the space, computing the distance to each of those points, and then computing a firing rate, how active each neuron is, basically by falling off with distance. So I'm going to choose some monotone decreasing function which modulates my firing rate corresponding depending on how far from the neuron's fixed location you are. And then I'm going to choose some threshold theta, and I'm going to say, okay, if your firing rate is above theta, then the neuron is on, and if it's below theta, it's off. So there's some background noise level or level of sensitivity that I care about. And because I have a metric space, looking at the pre-image of this the sort of sublevel set here, super level set here, is going to give me an open set, and it's in fact going to be a convex open set. So if I have enough neurons, what this gives me is a nice open cover of my topological space, corresponding to the activity of these neurons. So under very mild hypotheses, basically you just have to have enough of these things in sort of general position. Then you get an embedding of this space in what we'll call population activity space, which is to say I'm going to take all of my neurons that I'm going to observe in the neural system. I'm going to measure their firing rate, how active are they, right? That's a non-negative number. So now I've got sort of the first orthant of some large Euclidean space. The metric there is a little bit of a question, right? Neurons are definitely not independent, but we'll leave that alone for now. and so I have a copy of this space uh, sort of embedded in this funny way. And let me just pause right now and say I don't understand this embedding, and I don't know anybody who does. I think there's a tremendous amount of interesting work to be done just understanding what this kind of weird is sort of like if you take a manifold and you put a partition of unity on some open cover of the manifold, and we use that to embed it in a Euclidean space. The question is essentially what is the what are the in a nice partition with sort of this monotone decreasing property. You can ask, what are the extrinsic properties of this embedding? That's, as far as I know, entirely unstudied, but it's incredibly important in brain function for these brain regions. So just to toss a very large open question at your feet, I'm more than happy to talk about that with anyone who's interested and knows a lot more about geometry than I do. Okay, so neuroscientists, when they know what the receptive fields are, what the correlate is in behavior or stimulus, can then use the structure of these stimulus spaces to understand how population activity in the brain works, to make hypotheses. It's no mistake that two of the best understood areas in the brain are the hippocampus and primary visual cortex, and it's precisely because we have this really strong tell on what a large portion of the neural activity is going to do. So we can use this geometry to sort of reason about the system. This has not really been formalized in a mathematical way, the way that I would like to, since sort of bottom up. People tend to reason top down. But I claim that if we have, this is an embedded manifold, we should go get tools from topology, right? We have an open cover. We have everything we want. So let's apply topological methods to understanding these things. So, so how do I do that? So here's what data looks like, right? Here's what a neuroscientist is going to hand me if they say, okay, I've been recording from area X, and I should say this is a highly processed data set. So usually what they're recording is either electrical signals, what they call local field potentials around electrical probes, from which they then extract information about what neurons are spiking when through a very, very complicated series of tools, or they will nowadays use tools called optogenetics, where they genetically modify neurons to fluoresce when calcium enters the cell, which allows you to then use very tiny cameras to watch neurons be active. And then again, you have to extract all of this information from these very complicated videos. Everything is very noisy. It's very hard to get from there to this raster, which is just each row is a neuron, and time progresses, and those black ticks are spikes of that individual neuron. So each neuron is active or inactive. And you can see that, some of those neurons are active all the time. Some of them are sporadically active. There's a lot going on in this picture. Frankly, it's too much for me. So I'm going to try to understand not individual neural activity, but the population's activity. So I may as well not try to view these things in sort of in a vacuum, each individual neuron in a vacuum, but let's measure how activity behaves, or how each neuron behaves with regards to the others. So we'll introduce some symmetric notion of time series similarity. I'm not going to dive into which versions I like to choose, but the tools we're going to talk about today can essentially be applied to all of them. But the notion is I'm going to take all the data and I'm going to compress it into a nice symmetric matrix of n by n symmetric matrix, where n is the size of the neural population. So if I have a stimulus space, and here's an example, these are actual receptive fields imputed from actual neurons that we studied in the hippocampus for a paper in 2015. The Nerve Theorem, right, is this classical theorem from topology that tells you that the combinatorics of an open cover recover the topology of a space, but I can rephrase that in this neural activity sort of setting and say, okay, if I have a family of neurons and I know the receptive fields, the intersection patterns of receptive fields, I can recover the structure of the stimulus space, right? Same theorem, different words. So, okay, so here what I've done is I've picked out the centers of those three receptive fields. And you can see there's, some similarity and dissimilarity in the distances there. So the animal is going to move around in the environment, and the neurons are going to be active depending on how close I am to the target neurons. So if neurons are close together, then I expect the correlation between their activity to be higher. So that is to say there's sort of a swap here, right? What I want in order to think about the metric is distances, but what I've got is correlations. If there is a stimulus space floating around, then I should expect the correlations to be roughly inverse in order to the distances. And that is a giant, difficult pile of things to formalize. I'm not even going to try. I'm just going to say the intuition is that if you have similar activity, then you're more likely to have an intersecting receptive field. So what I'm going to do is go off to my applied topology grab bag, and I'm going to apologize right now to everybody in the audience who is not familiar with persistent homology, because this is going to be one of the shortest introductions you will see to this set of ideas, but I'm going to need to use it, and unfortunately there's just not time to dig in. But I will roughly say that what you do is you start with a finite metric space. Right, so in the top left, I've got some points floating around, and then I'm going to take a scale parameter, and I'm going to let the scale parameter vary. As the scale parameter gets bigger and bigger, the epsilon balls around my points in my finite metric space will start to overlap, and I'm going to start to glue in simplices, which are going to witness those intersections. So if I have two sets intersecting, I'm going to put in an edge. If I have three sets intersecting, I'm going to put in a triangle. Which is precisely constructing the nerve of the cover as the parameter varies. And then I'm going to compute homology of those sequential nerves. And I'm going to use the induced maps on homology to keep track of how cycles, what we call it, persisting. So it basically, cycles are born and then they die. So you get this nice decomposition as you do this, called the barcode decomposition, which is just a summary of the structure of the algebra, this giant pile of vector spaces, which are combined together to what we call a persistence module. And so here you see, the top set of bars there are Betty 0, which is just witnessing the sort of death of the connected components. The bottom set is Betty 1, which is witnessing the birth and death of one cycles of essentially, in this case, circles, because we're looking at a planar point set, as sort of there's enough edges to fill you in, and then eventually enough higher dimensional stuff comes along and fills you up. And so this gives you a measure of how, as I change a parameter, the structure of a space changes. But remember, back in my stimulus space model, I gave you a parameter. I said, okay, we're going to choose some threshold for distance inside of which a neuron is active. And since I'm using correlation as a surrogate for distance, what I mean now is I'm going to take high correlation neurons, and I'm going to give you some parameter that tells you what high means. And from that, I'm going to extract this combinatorial structure. I'm going to take that, compute homology, and give you back one of these barcodes. So that's our setting, that's our tool, and this is how we're going to study neural activity. And so according to the email, let's stop roughly 20 minutes in and ask for questions, and this is the end of the introduction, so that seems like a good time. So if anybody has questions at this point, I'd be happy to discuss them.
Hi,
Okay.
I've just got a quick question.
Yeah.
When you were explaining how you were embedding in a manifold, like to what extent do they already know that it is a manifold, or is this just part of the manifold hypotheses of, like, assuming that the data lies essentially in a lower dimensional manifold and then just trying to infer that structure of the manifold?
So
and like, how is it related to this toric result? Because I kind of got confused there, whether you were already trying to see it as being realized inside the torus, or whether you were trying to infer some other manifold structure. Thanks.
Sure. So the three stimulus spaces I presented at the beginning are sort of experimentally known from, a large body of work at this point. So everyone knows that simple cells in the primary visual cortex code for location and orientation. There's a lot of other cells in the primary visual cortex that don't do that. So the entire population doesn't necessarily have this coding scheme. And everything is stochastic. So it's a little bit like and I'm going to get way out of my lane and say something that I shouldn't. It's a little bit like the problem in physics where you have a space-time manifold, but if you look really, really, really close, it's definitely not a manifold down there, right? There's quantum physics, weird probabilistic things happening. If I look at neurons spiking and doing their individual events, this entire model doesn't make any sense to me anymore. It's a scale issue. So at the scale of neural receptive fields, in some systems, subpopulations of the neurons seem to code for things that look like geometry. I don't know if a manifold is the right tool here. It is the case that manifold models seem to do a pretty good job, but I would be not at all shocked to learn that if you look really close, that whole version, that whole story isn't a really great fit. But if if all models are wrong, some models are useful. We can still make predictions as though these things lived on a manifold, and we can try to study, the mathematical consequences for the coding properties of the neurons. In terms of the torus, so it was sort of the case that people saw this hexagonal lattice thing experimentally. These cells were discovered in 2010 or thereabouts, and they saw that they found the property of these things. It wasn't until mathematicians came along and said, oh, you can do this thing with a torus, that people said, oh, there's clearly a torus in there. But then it took another 12, 13 years for them to actually get good enough data and develop tools that were effective enough for us to actually verify the existence of this torus that we all mathematically conceptually knew should be there. So I don't know that you can say the manifolds are there in the sense that we mathematically might imagine them to be, but they do a pretty good job of predicting population activity for some subpopulations during certain situations. I hope that is clear.
Thanks. That's good enough.
Okay.
Those
Sure.
are very interesting answers. Thank you.
Yeah.
Ted, I had a very simple question for you.
Sure.
At the very beginning, you mentioned these monotonicity, monotone functions.
Mm-hmm.
Are these a model for physical mechanisms? Why monotonicity is taken into care?
Yeah, so that's a great question. So I use monotonicity to guarantee the convexity of the fields. That is demonstrably false, or it is just not true. Even if I just pop back to this picture here, you can see that there are places farther away from the center of the neuron where, from the center of the activity, where things pop back up. So there's a heat map here where red is highly active and sort of dark blue is not active. But again, all models are wrong. Some models are useful. And the other quip that I find useful here is that biology is nonlinear, but it's not malicious. I think the one, so in some sense, these neurons, if they are coding for a location, and it really looks like they are, then downstream neurons have to have some way of understanding that. And if I have a bunch of places where I'm, suddenly more active, even though I'm farther away, then the system becomes more difficult to understand, both for me, but also for the downstream neurons that are trying to interpret this coding, right? The rest of your brain has to figure out what's going on. So within some notion of, probability, there is some notion of monotonicity here that you might want to just take everything on that slide and put like a little expected value around it, and then cast it all in probability distributions to get a better version of the slide. But if I dive into that whole thing, we'll never actually get to talking about the results. You should take everything I'm saying here to be at best a rough approximation to the mathematics I really want to do.
For sure.
But yeah, I would say the monotonicity is a model assumption we make based on the fact that it almost appears to be true, and you could correct for that by taking sort of the long lifetime, or the long, if I imagine that in the limit of infinite data, and also if I happened to get the right stimulus space. Because what I've done in these pictures is take this data and compress it down into a two-dimensional space. But that's just because I know the cells are supposed to code for location, and they're supposed to do it, in this sort of two-dimensional space I know the animal is in. But maybe what I know, and a lot of neuroscientists tell you this, what I know is wrong. There's probably some higher dimensional structure. There's all sorts of other things the animal is doing with the hippocampus. It's also responsible for associative memory. So you could imagine there's a better stimulus space I don't know yet. And if I just knew it and I put these things in there, actually everything would be convex.
Well, sure, makes sense. Thank you.
Yeah, of course. Any other questions before we move on? Okay. I'm going to keep going, but please do feel free to hop in. If we don't get to the end of my slides, that's not a tragedy. Okay. So now so I will say we've been doing this for was 2012, so I've been at this for 10 years. Our initial question was just, can we detect the presence of a stimulus space? And we answered this question, yes, about 8 years ago. We used basically the tools I described to you just now to look at neural activity and say, okay, there is this kind of encoding going on. We can't tell you a lot about it because we're studying the topology of it, and you can get some geometry back if you're clever. But I would take this around on tour to the neuroscientists, and they were like, oh, that's very interesting. So how do we use it to decode what's going on in the brain? And so for the last, 7 or 8 years, I've been asking myself that same question. And so I decided to start with a feature that is pretty ubiquitous, which is the circle. So a lot of things in nature, right, are cyclic in the sense that they're periodic or they're well tracked by circular coordinates. And one thing that you definitely, we had that angular coordinate in simple cells and vision, but there are a lot of things you can think of that the brain keeps track of that are cyclic. One that I really like is head direction. So, if I just turn in a circle somewhere in my brain, there are neurons that are keeping track of what direction I'm facing, so I can close my eyes and do it, figure out when I'm facing forward again. So I must be doing it, right? Which means that there must be something in there tracking it. And those cells are detectable in the hippocampus and in other various areas of the brain. They're very commonly studied. Any pacemaker circuit, right? So my heart is beating at a fairly regular pace, probably a little faster during this presentation. But, there's a sequence of events that are happening over and over and over again. Any pacemaker like that is going to be well encoded by a circular coordinate. Fortunately, topologists are really good at finding circles. So, and computationally, this is the easiest place we can look. I will tell you right now that everything that we're going to talk about can be done for any dimension you like. If you really feel like looking for 7 cycles in the brain, go to town if you can make it work. But I'm going to give you examples in 1 dimension because those are easy to come by. So what I want to do is now, instead of just finding a circle, sorry, let me go back to my question. Now, instead of finding a circle, I want to find circles in 2 places, maybe in the stimulus and in the brain that I don't know are related, right? I don't have this dictionary of knowing what the stimulus space is anymore, right? My goal is to study it intrinsically. So I look out in the world and I, find a thing that happens to have circular features. I study the data and I discover the circle. And then I look in the brain and I probe around and I record a bunch of neurons in some rat or in, maybe myself if I'm feeling saucy. And I work out that there's some kind of circle in there and I can detect these things. So now I ask, is this the same as that? So, all right, so let's why is this hard to do? Why can't I just, I know I'm an algebraic topologist. There's induced maps on homology. This doesn't seem like it should be too rough. So, okay, so here's my favorite. This is a cartoon of an experiment. So there's a rat, and he's in a little circular environment, and he's going to run around and explore that track. And then I'm going to record neural activity while he does it, okay? So here are, the top is the barcode of the sort of the rat's location in the environment, sampled at some intervals. So I just take a bunch of points, and the bottom is the barcode these are both H1 barcodes, so I'm counting the birth and death of circles, like, circular features. And the bottom is the neural activity. So what do we see? Well, on the bottom, I see my circle that I want. On the top, there are two. And if you haven't seen this before, you would say, why are there two? There's one circle in the environment. Why is there a second feature? And it turns out that as the rat walks in a circle, right, walks around that circle, it's going to turn too, right? But it doesn't have to do that. Those two things are independent. It can stand in place and kind of wander around and look. And so as it moves around the circle, if it's not just running in a strict circle, then it's going to explore the entire surface of a torus, basically, exploring here in the corner and there in the corner, but turning around as it does so. So we get a pretty good map of the surface of a torus with that kind of exploration. So now the question is, which one of those things is recorded in the barcode in the brain, right? I just gave you some neural activity data. How do I know, is that keeping track of the direction the animal's facing? Is it keeping track of the direction the animal, right, the path the animal takes in the environment? Is it keeping track of both? Because they're naturally coupled. If it just runs in the circle, right, then it's going to turn in a full circle as it runs in a full circle. Or, and here's the really horrifying bit, maybe it's none of those things, right? It could be that I've just accidentally recorded from something that's keeping track of, the whisking motion of the mouse's whiskers or the heart or something else, right? So the problem is, I don't know what's there, and I don't know in particular that there should be a map. If I want to go to my algebraic topology toolbox and pull out maps induced on homology so I can hope to match these things, well, I better have a function underlying this, and I've imputed these spaces, right? I've imputed the two combinatorial spaces I'm working with from the data. So if I don't even know what the spaces are, where am I going to get a function? Like there's no hope of that, right? It's got to come from somewhere. So I've got to build something new that knows how to use something I can observe to match those things up. So here's what I can observe. I can observe a measure of cross similarity. Right, I've observed internal similarity between the animal's locations and built this combinatorial space. I've observed similarity between neural activity. I've built that space. But I've observed both of these things. So maybe I can take, for each neuron, I can say, okay, give me similarity to all of the things I'm measuring in the behavior space, to each frame in sort of where the animal is, or vice versa, right? I could say, okay, for each location the animal's at and direction it's facing, tell me something about what is going on in the brain. And so I could write down a measure of cross similarity. If these happened to be in the same metric space, then this is just the distance again. But if not, then it's something new, and that's okay. I don't have any particular requirement that this data have anything be calculated in any way related to the internal similarity between these things. Okay, so what have I done? I've measured the one thing I can hope to measure that gives me a notion between them. And if you look at this, you see a rectangular matrix, and if you're the kind of mathematician who looks at rectangular matrices, you might think that looks like a weighted relation. So it is, and I can construct something called the scale T Dowker complex of such a matrix. Dowker's theorem from the 1950s tells us that the reason it's called a Dowker complex is because he studied relations, and he came up with this notion of homology groups of a relation. And this is going to be, just like before, I'm going to take these measurements, and I'm going to threshold at various levels, and get sort of a sequence of inclusions of matrices and thus simplicial complexes. And if you've not seen the Dowker complex, I'm going to run us through an example in just a second, but just want to say this is the data you use to build one. So let me binarize. So here's a little example. So here I've thresholded, the black things are now ones and the whites are zeros, right? The black places are strong enough similarity. So what I'm going to do is I'm going to construct a simplicial complex whose vertices are the rows which is to say the elements of the first system that I'm interested in, and the columns are going to be the simplices. And essentially, you're going to take the downward closure of the list of simplices I get by taking, so for example, the edge between P1 and P2 is witnessed by Q1. Edge between P2 and P3 is witnessed by Q2. So I'll construct this complex, and I just realized I didn't change this, but there's a typo in that picture. The picture is correct, but I believe Q4 and Q3 have swapped places, so I apologize for that in advance. That triangle should be labeled Q4, and the edge at the top should be Q3. But this is the Dowker complex of this matrix. And you might say, but you said you could have measured things the other way, right? Similarity between distance, or between the input and the output, as opposed to the output and the input. So I could have done just the transpose, right? That should have been, there's no reason for me to choose one of these systems over the other. So if I transpose the matrix and do the same process, I get this other simplicial complex. And what Dowker observed was the following nice thing, which is that, well, I can take this thing called the barycentric subdivision of a simplicial complex, and when I do so, I replace, all right, my new vertices for each complex correspond to the simplices in the old complex, which is to say, the things I was using to index right in the top, I now have a vertex for each column, which are the rows downstairs. So if with just a little bit of fudging, this is not quite correct, but it's close enough, there's an embedding almost of one of these complexes into the other, and in such a way that it is in fact a homotopy equivalence. Recent work has souped this up to make it functorial, so you get this over the course of inclusions. So Dowker's theorem tells me that it actually doesn't matter which of these I chose to be the rows, which of the systems I chose to be the rows and which to be the columns, I'm going to get the same answer. Why does this matter to me so much? Well, one, because it's the data I can get, but two, because this provides me with a constructive way to take information, or to take a persistent homology on a complex whose vertices are P, right, one of my two systems, and turn it into persistent homology in a simplicial complex whose vertices are Q, the elements in the other system. So this is a bridge that I can use to take the Viator-Schrips complex, or the clique complex that I built using one of these dissimilarity measures, and turn it into information that looks like the other one. Not quite right, though. That's not what I wanted, right? What I wanted, sorry, let me hop back over here and just say, if we compute this thing, we get a barcode, and that's great. We've got the persistent homology of this new complex. Apparently we're sliding around a bit here. And I can apply Dowker to hop back and forth between something supported on vertices P and something supported on vertices Q, which is to say something about the mouse moving around its environment and something about the mouse in its mouse's neural activity. So this is the bridge. But it's not exactly what I needed. What I wanted was to compare the Viator-Schrips complexes, and what I've given you is comparison of this other complex, which wasn't either one of those things, right? I wanted to say, oh, I have a circular feature in this complex, and this other barcode, and I want to compare it to across the way. So what I've got to do now is build a method for getting to this thing from my original complexes. So right now what I've got is two different filtered complexes sitting on one set of vertices. And I've got to find a way, without any other real information, to tell you, cycles in homology on this and cycles on homology this should be matched up in some way. But the one set of vertices is a nice way to get your hands on things. What you can do, so here's an example. So here's one simplicial complex on one filtered simplicial complex on one set of vertices, and that hopefully is simple enough that we can all figure out what's going on. It's got one persistent homology class, right? So it's born at the second filtration and it fills in there at the end. And I could ask, okay, when I say that, when I talk about that homology class, that persistent homology class, what do I mean, right? It changes over the course of the filtration, but sorry, so I'll call it tau. And as it changes, I'm only adding things. It's a filtration, so I'm gluing things in. So as I move along that bar, right before it dies, it's that class is as big as it can possibly get. So those are all the things I could reasonably call tau, right? Every single, you can see there are several representatives of that class, and right where I've marked it there, that omega is where tau is at its biggest. So I have three distinct representatives, which I've listed there, and I would be within my rights to call any one of those things tau, I think. If you have more information, you might be able to say, well, some of those shouldn't be called tau. But I'm just looking at the combinatorics of the complex, and that there is this cycle in it, and it sort of lives. So that's what I'm going to do. I'm going to be as permissive as I can, because I don't know what I should throw away. I should say, well, if I want to compare two things, I should compare them in the most permissive way that I can. So those are the things I'm going to let you call representatives of tau. Here is my other simplicial complex that I've chosen to be as sort of ornery as possible. Right? We can see that now what I want to do is figure out whether or not there's a copy of tau living in there. So, well, it's got a nice bar, so that's good, and it dies in the same place. Maybe I should compare at omega. But you see, if I compare at omega, then there's a very good chance that I'm going to think these two things aren't the same, because I might, up top, choose that far right representative, right? I might say, okay, I'm going to go to omega, and I'm going to choose the smallest possible representative, because that's going to give me, right, a minimal generator. That's something we like in persistent homology a lot, because it gives us some notion of sort of tightness. If I choose that and try to map it down using the structure of the simplicial complex, I'm going to say, well, there's no match, right? Because that's going to map down, that little gold square is going to be bounded, and it's going to give me 0. So there's no match. If I'd gone the other way and chosen the far left cycle, well, then I'd gotten a good match, and the middle one wouldn't have mapped well at all. So in some sense, right, looking at, again, even though these are on the same vertices, sort of being choosy is not going to get me anywhere. I really need to be as permissive as possible. So what I'm going to do is I'm going to say, well, is there some representative of the class I'm interested in that appears as a representative in a cycle downstairs? That's the best I can ask for without adding any information to the system. So I'm going to find classes in the bottom complex, in the bottom filtration, anywhere that intersect the class tau upstairs, but I'm gonna look anywhere in the bottom filtration, because I don't know, that maybe for a brief while they matched and then they stopped. Upstairs, I'm gonna fix the largest thing, because I want to make that as permissive as I can make it. Okay, so I'm just gonna. So it turns out that I can ask this question in terms of a new filtration, which is to say I'm going to intersect those two complexes. So I take the first complex, and I fix the omega, the place I'm interested in, and then I just intersect it with every complex in the second complex, and I get this new complex. I can compute its persistent homology. It's got a little short bar and this nice long bar. But as if I add the assumption that the second complex is contractible if I let things go long enough, if I say, okay, eventually all homology vanishes in sigma 2, then anything that lives forever is just a homology class in sigma 1 omega, which is to say it's one of the homology classes I'm interested in. So I have a natural map now that I just look at the bars that live forever in this intersection, and that is something which is going to map to one of my original persistent homology classes. So all I have to do is ask which things sort of eventually end up on one of those bars, and that's going to give me the solution. Turns out that that's a linear system we can solve. Okay, sorry, I should have. This is the slide that says the thing I just said out loud. So I enumerate what we call extension elements in the persistent homology of the second complex that are in the image of those long bars corresponding to the cycle I care about. So here's an actual map on homology. I finally have it. I've got my hands on something I can use to answer my question. When are these two things potentially the same? I'm not telling you they are. I'm telling you this is like the most permissive notion of sameness I can give you. what I then need to do is run off to my collaborators who have domain knowledge and make sure that I'm not being crazy. But at the mathematical level, right, this is all I can do without adding information to the system, is sort of construct this whole thing. So okay, so now I've got a method for matching elements between two filtrations. If I have a bar that I'm interested in, a persistent homology class I'm interested in, I go nearly to where it dies, and then I can sort of look at all the things it maps to through these induced filtration. Okay, so let me put that all together. So here's my little cartoon again, right? The barcode I'm interested in. I want to know what that bar down in the brain is for, right? What is it doing? So I'm going to compare it to the witness complex using this induced filtration I just kind of very rapidly walked you through. So I'll extend it up to a bar up there, and in our little cartoon data, it absolutely matches to that witness bar, which is great because it tells me that there is some combination of things across, the relationship between the barcode of the witness complex and the barcode of the Viator-Schrips complex is not well understood. Those are open questions, so I don't want to suggest to you I understand what this matching is. We're working very hard on that. Very interesting questions. Happy to chat with people about it. But the method I just told you about tells me that there is an intersection of these two classes as sort of combinatorial objects sitting in this intersection complex. Okay, well now I can use Dowker's theorem, right, to flip it over to be an element of the other thing, and then I can do it again. And so now I get some other stuff up there. And I should say I'm sweeping a lot of algebra under the table when I'm drawing these bars because there's a choice of basis hiding in here. And also, you don't necessarily just get one answer, right? This is a relation. So there are actually a lot of things you match to up there. This is just sort of the primary one. And by primary, I mean something affine subspace offset. I won't try to go into the details because it's just way too much technical stuff. But I just want to say that's sort of what we would call the basic or the fundamental extension. There's an entire subspace of other extensions that you can hit, but those two bars are going to be involved. So that tells me that the thing in my sort of cartoon brain down here is that neural activity is witnessing the animal doing the sum of those two bars, and if I happen to know that the basis that I've chosen represents those two things as turning in a circle and walking around the environment, then this is the thing that gives me then the brain is witnessing the sum of those two things. It's saying, okay, you're definitely, if you just walked in a circle but you sort of intentionally turned so you're always facing the same direction, you wouldn't see this match. Okay. So great. So that's the tool, and I know that was very, very fast. I apologize. There's no way to do that quickly and effectively, as far as I can tell. Does it work? Now's a great time for questions.
Oh, given the context and the topic, is far too easy to make difficult questions to you and to guess. So what would be so the rat going in this circular pattern? Is it an experiment done when?
Okay. This sorry. I just so the rat going the experiment. So like I said, you should think of that as that particular experiment as a cartoon. So we did a lot of simulations and cleaning up to the point where the data doesn't really. But let's see, the data would have been. Well, I don't even know. It was. It's got to be at least 10 years old by now.
Right.
Data we based the simulation off of.
Any idea to repeat the experiment in a twisted torus, so to speak? In other words, a figure eight torus, almost flat, say, and presumably, I don't know how much of what you talked about would still survive in terms of information.
Yeah, so that's a great question. So because it's a cartoon, I'm not exactly sure how much of it would survive if we just like did a video of the rat and tried to make it tried to impute this from data. Like we really had to be pretty hands-on to get the exact setup. But what I will say is that the modern recording techniques are really good. In this particular toy example that I gave you, we don't know that, I can't say with any certainty what the survival rate of that toy would be into real data. What I can say is we've done this with real data. So we had simultaneous recordings from a lab in UCSD, Spencer Smith's lab, which is a microscopy lab. And what they do is look at they've got wide field microscopy of, they can get about half of a rat's brain all at once, or a mouse's brain all at once. And then they can do neuron level resolution in sort of an eighth of that. So I guess, what's that, a 16th of the mouse's brain, which is a tremendous amount of neural activity. But they can isolate individual regions in the brain and record the activity. So we looked at primary vision, where we knew that we would, have these circular coordinates. And we, sorry, excuse me. And once we'd isolated sort of V1, which is sort of the primary region in the brain, which takes input from the retina, a downstream region called area AL, which takes inputs from V1. They have an experiment where they showed a rat gratings, so these sort of repeating vertical and horizontal patterns. So what do we have? So area V1 and area AL, we were able to detect, even though so they only showed it sort of 4 angles. So we had horizontal, vertical, and then 2 others. So it was a very, very coarse circle. But using the methods I described to you, we were able to detect statistically significant Betty1 elements in both, persistent Betty1 in both area AL and V1 separately, and then get a good match between them, which is confirmation of known, coding properties of area AL. So without knowing anything about the stimulus, essentially we threw away the fact that the experiment was going on. We needed the experiment to happen in order to drive the activity. But just using the intrinsic activity, we were able to get a good match between the circular coordinates in these 2 brain regions. So we do have proof of concept in actual neural activity, which is fantastic. There is a preprint which talks about the mathematics. I will say we haven't even begun to delve into the actual, theorem proving here. We really need to understand this whole, we proved that everything does what we say it does. We have not proved anything about how well it does those things. We know from sort of general nonsense that you'll get a stability of representation, but we don't really understand the geometry. My current postdoc, Nico Schoncheck, is working very hard on developing sort of, we're going to sort of try to lift this to a notion, sort of a continuous version of everything I talked about, so that we can start proving convergence properties instead of working in simplicial complexes where it's very, very hard to think about those things. Okay, so the thing that we're asking now is, so now I know that I can check from observation of activity whether or not 2 circular coordinate systems are, quote unquote, the same, or at least could be the same. And so what we're hoping to do is to apply that technology to ask the following question. If I have 2 neural populations, and I train one of them to represent a circular coordinate, how do I train the network to pass on that circular coordinate and not, say, some other random circular coordinate, right? If I give you a random network, how well does it pass on circular coordinates? Is this a generic feature of neural networks, right? Do they always sort of permissively let you transmit this information downstream? And if not, if I say my input is sensitive to an 8 torus, right? I've got a whole bunch of circular coordinates faithfully represented, and I want to pass information down so that neural processing can happen. Well, I don't just want to dump all that information. The brain is very good at sort of segregating data into individual pieces, doing computations, and then reintegrating. This is sort of the large-scale computational picture we have of what the brain does. In that setting, how do I how do I select? Okay, I want that circular coordinate and that circular coordinate, or maybe the sum of those two circular coordinates, but I don't want that one over there. Let's not hand that one down because it'll interfere with the computation. So, so we're currently the last set of slides that I was going to show you after the proof of concept slides, which shows the computer, is that we're sort of investigating what neural architectures provide you with that sort of permissivity, and how you would learn, what learning rules are effective, local learning rules, the kind that the brain could implement, where they say, okay, I'm a neuron, I don't have an overview of the fact that you care about circles, but what can I do to learn to pass on your circular coordinate system that you care about? So that's work still very much in progress, but we've observed some very interesting things about random networks sort of doing a good job of passing along a lot of pieces of your circular coordinate, but they happen in this scrambled way, which you might expect from a random network. And you can then nudge them to give you different versions of the upstairs circular coordinate. I really wish I had my picture, but it seems like you're very close to making many matches in a random network, which is fantastic, right? It tells you that it's easy to learn circular coordinates, even if you don't naturally have them, but they aren't intrinsically present. And this is the best thing you could hope for, I think. I should say that's preliminary work. We are still very much trying to understand it, but it seems like we have the ability to use this tool to infer some things about neural architecture. So that's very exciting. And I think I'm out of time. So let me open the floor to questions, and also flee from trying to talk about things without slides. So thank you.
Chad, thank you for a very interesting talk. Have you thought any about looking at other kinds of brain functional data, like in humans, like positron emission tomography, that sort of thing, where you've got voxel information rather than individual neurons?
Yeah, thank you. So that's a great question. So we did some work with this when I was at Penn, where we looked at, so not functional data, but structural data, diffusion spectrum imaging, which gives you sort of a picture of the white matter tracts in the brain. Yeah. And the reason that we did that is largely because that data is much easier. But we found in that setting, we actually did find quite a lot of structure in the clique complex, the weighted clique complex of the network, which matches, some of the cycles that we found. We found a two cycle, which looks a lot like the so-called what-where pathway, which goes from primary visual cortex up through up to the front of the brain, but it sort of it segregates data, like I mentioned earlier, into sort of two pathways. The what pathway, which goes through where pathway, which goes through primary motor cortex, which lets you get out of the way of an oncoming car without really thinking, and the what pathway, which lets you recognize faces and do complex processing. So there's a much faster pathway. Right, so you take the primary data and you sort of, you blow it off in two different directions, and when it comes back together, you know who you're talking to and where they're standing. But those require two different paths. So we found something that indicates that looked a lot like that. MRI is much harder, and so PET is much harder because the data at the voxel level is sort of imputed through these inverse problems, and it's very, very hard to trust any given measurement. And so again, you have to find the right level of abstraction. I think we worked sort of at the level of what people call regions of interest, where you take maybe even a fist-sized chunk of brain, you average. That whole thing destroys so much signal. We never really nailed it. I do think there are some groups that are doing this, and I apologize off the top of my head, their names are escaping me, but if someone is interested, I'd be happy to remember those and email later. There are some groups who are really trying hard to study, like, for example, are there cycles at the voxel level that you can detect? Right. So that's ongoing.
Okay, if there are no others, then thank you very much, Chad. Terrific talk.